we will move on to part 2 from chapters 8 and 9. Remember, in part 1 we looked at evolution that works at more than one locus for a trait. So if you have particular traits that are multi-loci, how is selection, drift, etc. going to work on those kinds of traits? What we're going to do now is we're going to look at the evolution of sexual reproduction. So remember, at the end of last time we talked about one of the important things with sexual reproduction is that it is one way that we can reduce or eliminate linkage disequilibrium in a population. So with that in mind, this next part looks at the evolution of sex and the adaptive significance of sex. There's a lot of different types of reproduction, and sexual reproduction, where we produce haploid gametes, is only one of those types. So part of the paradox of sex is that it essentially reduces an individual's genetic contribution to the next generation by 50%, say if we compare it to a clonal organism. So if I clone myself, 100% of my genes would go into the next generation. If I reproduce sexually, only 50% go into the next generation. And so one of the ideas is that that is, of course, hugely costly from a fitness perspective. The other side of the coin is that sexual reproduction is probably far more common in nature than asexual reproduction. So even though there's huge cost, it is very prevalent. And so that's where this paradox comes in. You've got something that's highly costly, yet also very prevalent. So what is the benefit? If the cost is that high and it is so prevalent, there must be some very large benefit to sexual reproduction. So one of the early folks that looked at this was John Maynard Smith, and Maynard Smith was a mathematician, biologist, and so he came up with some models, and one of his early models was, as you see here, there's a couple of assumptions. So first off, one of his assumptions is that a female's reproductive mode does not affect the number of offspring she can make. So we're going to be comparing essentially an asexual female who's cloning herself to a sexual female who's reproducing sexually, and we're gonna look at their fitness. And so the first thing here is the number of offspring that each of those two can make is going to be the same. So they're not going to make different number of offspring. They're going to make the same number of offspring. And then the other thing that assumption that's here is that whether she's sexual or asexual does not affect the probability that her offspring will survive. So she's got the same number of offspring and equal likelihood that those offspring will survive. So essentially they're equal in all ways except that one is sexual and the other is asexual, but everything else we're holding constant. The other thing that we're going to look at is if you are a sexually reproducing organism, then when you go to make offspring, half of them will be males and half will be females, whereas in an asexual organism there isn't another sex, so they're all just, in this case we'll just call them females, but essentially that's what we would look at. So there's not going to be differences in the sexes there. So what this is trying to do is to show us that the asexual females are really going to have an advantage. So we think of this asexual female, and she's going to have a certain number of offspring, and this asexual female over here is going to have the same number of offspring. So remember, our assumption said they had the same number of offspring, but it also said that over here in the sexual, there would be 50-50 males and females. So what we look at here then is we're asking what is the proportion of asexual individuals to sexual individuals in the population. So here we had this asexual female had 4 offspring. Here we had this sexual female mated with a male also had 4 offspring. So in the beginning, our asexual individuals were 1/3 of the population if you just count all these individuals up. By generation 2, they're actually 1/2 of the population, and then by generation 3, they're actually 2/3 of the population. So what you're seeing is increase in the number of asexual individuals as far as the frequency goes, and then a decrease in the frequency of sexual individuals. So essentially, any genes that would be causing that asexuality have a higher fitness, and they are increasing in frequency in this population. And pretty soon, what you would expect to see here is everyone in the population would have this asexual gene, and we would eliminate the sexual gene. So there's an experimental model that's been used to test this. And this is a picture of a nematode, and nematodes are roundworms that DNA sequence has been well sequenced, and a lot of the functions of some of those different genes are known. They've been used a lot for different studies and lots of studies on aging and this sort of thing, the value of sexual reproduction. So what we're looking at here, remember in sexual reproduction you get recombinations, you get new combinations of traits, whereas in asexual reproduction you do not get those new combinations. Your offspring are going to have exactly the set of characteristics that you had. You don't get new combinations of those. And so one of the things that this is showing us over on the left hand graph is that first off what they did is they took two populations, and one of them is a control, the other one they exposed to a mutagen, so they caused mutation to occur. And what you're seeing here is that especially if you compare the mutagen group with the control group, one of the things that happens in this mutagen group is that it's doing a lot better if it can have a significant amount of outcrossing. Okay, so outcrossing would essentially mean that it's mating with other individuals. So it's not just selfing. So in this type of organism, they can fertilize themselves or they can fertilize other individuals. And so if they fertilize themselves, then this outcrossing rate is 0. If they basically never fertilize themselves, they only fertilize other individuals and become fertilized by other individuals, this outcrossing rate is 1. And so what you're seeing here is the real advantage to outcrossing definitely occurs in these populations that are exposed to the mutagens. So if you are a population that's experiencing a lot of negative environmental influences, then outcrossing is definitely to your advantage. In all cases, the control population did better than the mutated population, but where the mutated population found its greatest benefit was if it had some level of outcrossing. We expect then, if you're in a highly variable, stressful environment, that outcrossing perhaps is beneficial and increases your fitness. Then what they did over in the right-hand graph is they took some different populations and they looked at the outcrossing rate across the different generations. So let me take populations, and in one set of populations I'll expose them to mutagens, and this other set of populations will be my control. I'm not going to mutate that line. And what you're finding is, if you sort of look at the difference between the two graphs, is that over time the population that was exposed to mutagen switched to a slightly higher outcrossing rate, right? Because remember, outcrossing helped those populations to do better. And so if I continually expose a population to mutagens over time, it will increase its outcrossing rate. So again, this is evidence that suggests that selection is favored, or that selection favors higher rates of genetic recombination in stressful environments. And that makes sense, because if you recombine your genes, you might be able to get a new combination that is now better in this to handle this stressful situation. Whereas if you just clone yourself, your offspring are going to have the same level of resistance to whatever these stresses are that you had. There's not going to be anything new. One of the other things that they talk about is how sexual reproduction can help to combat the effects of mutation and genetic drift. So what we're looking at in this particular figure is that we've got a population, and we start off with a histogram that looks at, in this population, how many individuals have no harmful mutations, how many have 1, 2, 3, 4, 5, etc. So at the beginning of the population, it's pretty evenly distributed. On average, they have about 2 harmful mutations is the most common, and then only a few individuals have no harmful mutations, and a few individuals have 5 harmful mutations. So most individuals are right here around 2 and 3 in the population. Now what's going to happen is we're going to take this population and we're going to allow it to get a mutation. Okay, and so basically what we're going to do then, once we increase, let's say we start mutating it, then imagine, let's say that these individuals here who have 0 mutations, they mutate. So now they don't have 0 anymore. They sort of jumped over into this 1 group, and then maybe some of the ones over here in the 1 jumped into the 2 group, etc. So by inducing a mutation, we're sort of moving this entire graph, sort of shifting it to the right each time. So now most individuals actually have 3 harmful mutations, and if we keep doing that, we're going to get it to shift again. So we're not going to get back this 0 class, right? Because our previous population had nobody in it that had no mutations, so we're not going to come back to that. And then when these individuals that have 1 mutation, when they get a new mutation, we're going to lose that group as well, and we're not going to come back to that. So again, over time, this whole thing is going to shift to the right. This phenomena is called Mueller's ratchet, and think of it like a ratchet wrench. What that does is it's something that can move in one direction, but it can't go backwards. And so in asexual populations, you tend to accumulate more and more of these harmful mutations, because remember, they're not going to be able to get rid of them through sexual reproduction because they're asexual. So once a clone has a mutation, all of its offspring after that are going to have that particular mutation, plus any new ones that happens to arise. Right? And so each clone that comes after is just going to be the accumulation of all of the different mutations that have happened before it arose. So this is what we're seeing here. Now, sexual reproduction can break the ratchet, because what would happen is in sexual reproduction, if you have individuals that have, say, let's say you have some individuals that have 1 harmful mutation at one place, and then you have another parent that has a harmful mutation at another place, through recombination, you actually could get an offspring that didn't have either one of those mutations through recombination. So they didn't get the bad mutation from parent A, and they didn't get the bad mutation from parent B. They have a whole new set, and they now have no mutations. So sexual recombination can get us back these groups that we lost. But sexual reproduction, we will not get those back. We will just continue forward with those increasing the number of mutations. Again, we're asking, what is the adaptive significance of sex? So we know that sexual reproduction can eliminate some of those harmful mutations in a population that we saw in the previous slide. One of the other hypotheses that's out there is called the Red Queen hypothesis. And the Red Queen hypothesis specifically deals with this evolutionary arms race that you have between hosts and parasites. And so what this is doing is it's basically placing this host-parasite relationship at a very prominently featured place in evolutionary history, because it's saying that it is likely that sexual reproduction evolved as a response to hosts and parasites dealing with one another. So an arms race is what you would expect, where essentially in this case what you have is you have an adaptation that occurs in one of the individuals, let's say the host. So let's say our immune system gets better at detecting and warding off this particular parasite, but then there's a mutation that occurs in our parasite that actually is beneficial and allows it to overcome our defenses. So now, of course, those individuals increase in frequency in that population. And now what's going to happen in our population is that any individuals in our populations that has a mutation that can overcome that particular parasite is also going to be beneficial, so that will be selected for. So basically what they're doing here is thinking about, what if you just had 2 different genotypes in a population? So let's just say you have susceptible and resistant, right? So rather than looking at all sorts of different possibilities, you just have 2 different ones. And so what you're looking at here in B, you've got this host population. So you either have high resistance to pathogen 2 or you have high resistance to pathogen 1. So there's a trade-off because you don't get to do both. So you can have one or the other. And so what they're saying here, if we look down and see, this is where that perpetual arms race is. So if in the population we have a whole bunch of parasites that have genotype 1, we're going to select for resistance for genotype 1 over here. And then what's going to happen is that then favors in our parasite an increase in genotype 2, right? Because now they're doing well. Genotype 1 parasite's not doing well because our immune systems are fighting it. And so now genotype 2 parasite does better over here. And so what that does is then, now that there's a whole bunch of genotype 2s, now we as the host are under selection to favor genotype 2 because they're going to do better. And so essentially this cycle just continues. And so the idea is, since there's not really a fixed pathogen resistance level, that here sexual reproduction is better because it allows the population to keep that variability. It allows us to have in our population both resistance for pathogen 1 and for pathogen 2 in that population. And so it allows us to or it's beneficial for organisms to have sexual reproduction so that their offspring, occasionally they can make offspring that would actually be resistant to this new parasite. So here are some data that are consistent with the Red Queen hypothesis from trematode parasites. So these trematode parasites essentially get into snails and they change the behavior of the snail. And one of the things that you'll find is each one of these pens right here, these pie charts, this is looking at a separate population. And then what the pie chart is showing us is what percentage of that population is actually male. So if it was something that is completely solid, that's a completely asexual population right there. There are no males. It's just females. And then where you do see some white, they are reproducing sexually a little bit. They're producing some males, so there's some sexual reproduction. So you can see over here the male frequency is anywhere from 0 in these ones that are completely solid to almost about, say, 0.35 in some of these ones that have a larger white area in them. So some populations are producing about 35% males, and then there's lots of populations that aren't producing any males. But what they're doing also is they're correlating that with the infection rate. So this infection rate is essentially looking at the level of infection by these trematode parasites. And what you're seeing is a very nice positive correlation between the rate of infection and male frequency. So remember we said before that sometimes we think sexual reproduction is a mechanism that has evolved that allows organisms to respond to variable and stressful conditions. And that's what we're seeing here. So what we're seeing here is that those populations that have a lot of infection through these parasites, they actually are in most cases producing more males, and i.e. that means they're switching to sexual reproduction. So as the infection rate of the parasite goes up, the amount of sexual reproduction also increases. Here's a video that is really not related to sexual reproduction, but it's a kind of a cool video that shows how some of these parasites get into the snails and actually change the behavior of those snails. And it talks about other parasites as well, how they can get in and change the behavior of their hosts, and they do it in a way that makes it so that their host is more likely to get the parasite to the next host it needs to go to if it's a multiple host system. So it's a pretty interesting short little video there. Okay, so the next thing we want to look at is quantitative genetic analysis. So in the past we just looked at, basically we have a locus or maybe even two loci, but we had very discrete types of traits. Brown eyes, blue eyes, brown hair, blonde hair, you are a taster, a not taster, that kind of thing. Very, very discrete. Whereas in many cases, what we find in nature is not discrete. It's not like you're short or tall. There's a level of different heights that you have. And so those are referred to as continuous distributions, and those are called quantitative traits. So discrete qualitative traits would be, again, something like male versus female. If you had 2 different color morphs, or even 3 different color morphs, that would be a discrete type of a trait, and we would also call it qualitative. If instead you had, many, many different heights in a population, many different running speeds, all that kind of stuff, then those are quantitative traits, and that's a continuous distribution. And it's considered continuous because essentially you have an underlying continuous number line down here, and data points falling almost any different place, at least within this continuum. So we don't just have these big buckets. You can fall pretty much anywhere in between there. So one of the things to think about, if you go and you look at a population and you're looking at a particular trait, and that trait shows this type of continuous distribution. So you measure all the lizards in your backyard and you measure their tail length, let's say, and what you find is you have all these different tail lengths. And so the question then becomes, if you find that phenotypic variation in a population, then what is it that explains that? And really there's going to be two major things that's going to give us that type of quantitative nature of a trait, that continuous variation. And it could be both of these. It could be one or the other or both. One of the things could be that you have a trait that has lots of different loci. So if you have just, as opposed to having just maybe one or two loci that are contributing to this particular trait, you have lots of them. The other thing is you might have a trait that has a lot of environmental effects, and we'll see that here in a second. So this is looking at traits where you have multiple loci for these and how that affects quantitative traits. So if we look over here on the far left, we can see that if you have one locus, in this particular case, the best you're going to get are 3 different phenotypes. So this one would be, let's say, an individual that has 2 recessives and it's short. Here on the right, we have an individual that has 2 dominant alleles, and so it's tall. And then maybe this individual here in the middle has 1 dominant and 1 recessive allele, so it has an intermediate height. Okay, and that would be basically incomplete dominance if that were the case. What is showing here in the middle is what happens if you expand that to 2 loci. So if we had 1 locus, we had 3 possible phenotypes. If we have 2 loci, you can see here we could get maybe 5 different possible phenotypes. And really what they're doing here is looking at the possibility of what we sometimes call additive effects. So the shortest individual here would have all recessive, and then these individuals would have maybe 1 dominant. So of the 4 different alleles it would have, one of those is dominant. And then the one in the middle would have 2 dominants, and then 3 dominants, and then 4 dominants. So it doesn't really matter whether it's the A or the B. It's just essentially what they sometimes call dosage. It just looks at how much of each or how many dominant alleles they have, irrespective of which gene it is. The more loci you add, so this is looking at what happens if you have 6 loci. Again, think of it as dosage things. So all recessives, and then you have 1 dominant, 2 dominant, 3 dominant, 4 dominants, etc. So of course if you go across there, you have a lot more different types of individuals. So the more loci you have, the more continuous it becomes. And then over here they show us just sort of that's the model. Now we look at a particular set of data. Now the second thing that can give you this continuous distribution is if you have environmental effects. So even if you took a plant that genetically is identical, so we're going to take a plant that is a clone, and in this particular case what we're doing is we're taking this cloned plant and we're growing it at different elevations. So genetically there's no difference there because they're clones. But if we grow it at an elevation of 100 feet, it grows very, very tall. At 4,600 feet, it's sort of intermediate in stature, and then if we grow it at 10,000 feet, it's very, very stunted. So there's not a genetic difference there. This is all due to the fact that they were raised in different environments. So again, if we went out and measured this population, we might see that if we went across an elevational transect, let's say, we would see lots of different heights of plants, but it wouldn't be because they're genetically different. It would be because are these environmental components that are influencing that phenotype. Now your book talks a little bit about quantitative trait loci and how we go about mapping them. We're only going to talk about this for a few minutes, and you don't have to worry about all the detail that's in the book. Basically what they do is they take, and let's just say that we have a particular trait that we know is being coded for by lots of different loci. So there's large portions of the genome that contribute to this trait. Then that we refer to that or those portions as quantitative trait loci. So if you could just go and identify all these different genes on all these different chromosomes and sort of group them together and say these all go into making up, let's say, IQ, if you will, then we would say that those are the quantitative trait loci for IQ. So we do know that there are some of these widely spread traits that have a lot of different genes that contribute to them. So when we try to map those, we refer to this as QTL mapping, where we go and we try to actually look for some of these different loci that contribute to some of these quantitative traits. And this is just an example. One of the QTLs that they've been looking at in humans, we have novelty-seeking behavior. And it appears that this is a personality trait that is more common in individuals depending on the type of dopamine receptor they have. So in essence, you can see you can either have a short or a long receptor, locus receptor. And so these are the receptors for dopamine. So dopamine is a chemical that's released that provides, when it binds to particular areas of the brain, the pleasure centers, it gives you a feeling of pleasure. And so if you have more dopamine receptors in certain parts of the brain, you're going to have that higher reward system being activated. And so what you can see here is individuals that have both SS copies have a much lower, if you see here 55.1, a much lower novelty-seeking score than individuals that have either 1 or 2 copies of the L longer tandem repeat. So there appears to be some correlation there then between these QTLs that are out there and novelty-seeking behavior.